And now is it audible to you, sir? I will share my screen, sir. I will share my screen once again. Yes, sir. Yes, sir. Absolutely. Absolutely. Yes, sir.
Sir, good morning to all of you. Welcome to the fourth day of 1 week faculty development program. And Dr. Nagaswamy sir, welcome sir. And thank you for accepting our invitation as a resource person of the fourth day for the faculty development program.
Good morning, sir. Thank you very much for your kind invitation. It's my privilege
thank
to
you for all the participants. Now I am introducing our resource persons of the today's program. Dr. D. Nagaswamy Venkateshan. He is from JSS College of Pharmacy from Tamil Nadu. Presently, he is working as an assistant professor in the JSS College. And sir has published a number of papers and book chapters, 65 research papers, 35 review papers, and 4 book chapters. And he filed also patent for one. A number of conferences also attended, that is 40 number of conferences had attended the sir. And he delivered 20 lectures in the various national and international seminars and presentations given. And he acted as a chairperson for the 2 national and 2 international conferences. And for 6 international conferences, he presented the papers. And he also acted as an organizing committee member for the 20 international conferences. And he is also an advisory board member for the 3 national conferences. And presently, the sir is working as a assistant professor in the JSS College of Pharmacy. Thank you, sir. With this brief introduction, once again, I am welcoming you.
Thank you so much for your nice introduction. Good morning, everyone. Am I audible to every one of you, please? Am I audible, sir?
Yes, sir. Your voice is audible, sir, for me.
My voice is
okay,
clear.
Sir.
Too?
Yes. Okay, everyone is saying yes through the messages, no problem. Share your screen and you can start, sir.
Is my screen is visible to you, sir? Sir, is my screen is visible to you?
Yes, sir. It is taking buffering. Maybe 1 minute. Yes, sir.
Can we move on to that, sir?
Yes, sir. Okay, okay.
Good morning, my dear fraternity colleagues across the country for taking part in this FDP program. It was a very good introduction given by the sir. So the topic for my presentation today is phytosomes, a promising approach for the delivery of phytoconstituents. Just if you look at back the olden days or few decades before, Herbs and medicinal plants are known for millennia, and they were highly recognized as rich source of therapeutic agents for the treatment, prevention, and cure of diseases and ailments. These medicinal plants and herbs are the ancient remedies which are used by the mankind, especially our ancestors and elders. So the advances in herbal drug has gained a recent attention in treating the diseases, and among them, the phytoconstituents are gaining very much attention these days. Over the past few decades, if you look into the research activities, it was mainly concentrated on the plant-based research to identify a new chemical composition for certain new indications to treat the diseases and ailments by the traditional medicines. Till now, approximately 4,000 and odd naturally occurring materials have been identified, which has the diverse pharmacological effects, which are predominantly flavonoids. And as per the WHO, about 90% of the inhabitants in this planet they rely on the plant products. However, India is well known for its traditional knowledge of using the medicinal plants for variable living traditions. So recent informations on medicinal plants indicates that it has a growth of 12% annually. In India, if you look at the medicinal plant value, it estimates around 7,000 crores with an export volume of 600 crores nearly. But compared to the global market, our Indian market contribution is likely to be low. So why this has happened? Since long time, these medicinal uh, herbal medicines were not considered for the novel formulation. Why so? The reasons are lack of scientific justification, and we can find many processing difficulties in the formulation, which includes the extraction of material from the plant and identification of the individual compounds creates problem in the herbal formulation. And apart from that, the separation and purification of the medicinal plants may lead to the loss of the plant. So if you look at the biologically active constituents, that is phytoconstituents in the plant, which are highly polar in nature and water-soluble molecules. To name few, we have flavonoids, tannins, and aglycosic glycols. The problem associated with these phytoconstituents, when they administered through oral route, they exhibit poor absorption, which happens due to high molecular weight. Recently, there is a paradigm shift in developing a novel drug delivery system for delivering such phytoconstituents, which includes uh, various drug delivery systems such as nanoparticles, microemulsion, matrix systems, solid dispersions, liposomes, and as well as phytosomes. So, moving on to the further on phytosomes. So this is the contents of my seminar, phytoconstituents introduction, phytoconstituents advantages, disadvantages, introduction to phytosomes, and we have components of phytosomes, role of phosphatidylcholine in phytosomes, the preparation techniques in phytosomes, formulation of phytosomes, characterization of phytosomes, and the difference between the phytosomes and liposomes, evolution of phytosomes, and some of the recently published articles where they have used a quality by design approach for the phytosomal formulation, marketed formulations of phytosomes, and patented phytosome formulations. So, from year 2017 to 2022, if you see the phytochemical market, it has given a contribution of 5.6 compound annual growth rate as compared with other markets. So the market value, if you see, it is US dollar 7,436 million was contributed from the phytochemical market as compared with other traditional medicines. So the overall growth was 7.1 percentage compounded annual growth rate. So if you see the different countries where these phytochemicals were predominantly used in their formulations are among the top, we can find China, which contributes around 10%, and this is followed by India to an extent of 6.2 percentage, and USA 4.7 percentage, Germany 2.1 percentage, and Europe 2.8 percentage respectively. So when you see the global phytochemical market worldwide, as on 2022, it was nearly 5.6 percentage, and it has grown up to 7 percentage in the year 2023, and it is expected to grow around close to 14% in the year 2033. So this is a growing phase where it indicates more research has been devoted in the development of phytoconstituents. So already discussed, these phytoconstituents are nothing but they are the preparations which are derived from plants or part of the plant which have been used from ancient times for the use of certain diseases and ailments. So medicinal plants have been known for many decades and they have been widely recognized as high therapeutic agents with numerous health promoting benefits for the treatment of diseases and as well as the ailments. Over the past century, the chemical and pharmacological sciences established the composition, identify the biological activities, and health giving benefits of numerous plant extracts. It is often observed that the separation and purification of the various components of a plant extract, which may lead to a partial loss of the specific activity of the purified component. So this is one of the disadvantages we are facing with the phytoconstituents. And if you look at the phytoconstituents, mainly the phytoconstituents are flavonoids, terpenoids, and polyphenols, which are likely to be high polar in nature, exhibiting high water soluble molecules. And these water soluble constituents are poorly absorbed due to their poor lipid solubility, which hampers their ability to cross the lipid rich biological membranes. Ultimately, which also results in poor bioavailability in the systemic circulations. Many approaches have been developed so far in order to improve the bioavailability of these phytoconstituents, which includes the enhancing the solubility and incorporation of bioavailability enhancers and modifying the structure on or entrapping into a lipophilic carrier. Therefore, what happens is the chemical complexity of the crude or partially purified extracts seems to be essential for exhibiting the bioavailability of the active constituents. Some of the water soluble extracts, upon oral administration, may face some kind of issues, which includes the destruction in the gastric environment and destruction by the gut enzymes. Recently, many research has been devoted for developing a drug delivery system to overcome the problems associated with the herbal medicines upon oral delivery by a scientific approach And this approach includes the development of a novel drug delivery system. So this is a novel technique where we can improve the oral viability of the phytoconstituents for the medicinal preparations. So the advantages of the phytoconstituents are they are obtained from the natural sources, and they are rich in pharmacologically active products. One example I wanted to give here is alkaloids. They are abundantly available. They are biocompatible, does not exhibit any incompatibility with other ingredients which are incorporated. They have improved potency and safety, and they exhibit as a lead molecules for the discovery of new chemical entities. So these are all the advantages of the phytoconstituents. Despite these phytoconstituents have excellent bioactivity in the in vitro models, they exhibit less or poor action in vivo model. So these are all the different disadvantages are often associated with the phytoconstituents when they are given in the conventional system. So major limiting steps with the phytoconstituents are they exhibit poor lipid solubility, and they have improper molecular size, and this improper molecular size finds it difficulty in crossing the biological membranes and entering into the systemic circulation when given by oral route and as well as by the topical administration. So they exhibit high polarity and the water soluble nature, that is polar nature. Examples, these flavonoids, tannins, and acetoacidic glycolides that restricts the absorption of the phytoconstituents by the passive diffusion process. Along with that, some of the phytoconstituents, they have long side chains and high polar in nature that can also render problem or difficulty in pass through the lipidic skin by passive diffusion process. And I already told in the introduction part, destruction by the gastric environment. And so the gastric juice and as well as the gut enzymes can also have a role on the phytoconstituents to get degraded in the system. But when often individual components were separated from the whole plant, they lose their activity and the synergistic effect of the phytoconstituents were also lost. And other than that, we have the chemical complexity of the crude or partially purified extract to be essential for the viability of the active constituents. And some of the water-soluble extracts upon oral administration may get destroyed in the gastric environment. So these are all some of the disadvantages. So how we can overcome these disadvantages or drawbacks? So the recent years, there is a tremendous scientific effort has been devoted for the development of the novel drug delivery system for the plant drugs or plant extracts. So such a system includes the development of polymeric nanoparticles, nanosomes, transferosomes, pharmacosomes, solid lipid nanoparticles, and phytosomes. These drug delivery systems have the ability to enhance the rate of release and ability to cross the lipid-rich biomembranes. So what happens is a novel phospholipid, phytophospholipid complexation technique is commonly named as phytosomes, which plays an important role in increasing the absorption and improve the bioavailability. And also some of the phytopharmaceuticals which has difficulty in solubility and as well as inability to cross biological membranes. So phytosomes can solve these problems by enhancing these two properties. So later the science has moved forward and they have discovered that a complexation with other chemically useful nutrients, namely these phospholipids, emerged as one of the important component in the development of phytozoans have improved the bioavailability of plant extracts or phytoconstituents. So these nutrients are also helpful in enhancing the absorption of the phytoconstituents or phytochemicals. Phospholipid-based drug delivery system were found to be an effective and successful drug delivery for the phytochemicals. How do they success in delivering these drug delivery system? These phytozoans, they are resembling like cell membrane with the building blocks making up of matrix which can able to accommodate large variety of proteins, enzymes and biologically active converters. In humans and animals, these phospholipids, besides they are having the role as a carrier, they can also help in the digestive aid for both water miscible and water fat miscible nutrients. So coming to the introduction part of the phytozoans, the term phytozone denotes phyto means plant and somas means cell-like structure. And this technique was developed by Indana Spe, Italy. They are the um, um, inventors of these phytozoans. And if you could see this technique, you can find many papers developed by the Indana Spe. The group of scientists they have identified that incorporating phospholipids into the standard extracts so vastly improve the absorption process and utilization of the phytoconstituents. These phytoconstituents they have a cell-like structure, a novel approach to drug delivery system which can overcome the problems associated with the conventional drug delivery systems. So, in phytozoans, the bioactive phytoconstituents or the phytochemicals of the herbal extract is surrounded by a lipids. And apart from that, the phytozoans, they themselves act as a small cell and render protection for the valuable components of the herbal extracts being protected from the destruction by the digestive secretion and as well as the gut bacteria. What happens exactly? The water-soluble phytoconstituents can be converted into lipid-compatible molecular complexes that facilitates herbal extract to pass through the lipid-rich biomolecules. However, these water-soluble constituents, namely flavonoids, tannins, and aglycolic glycosides, are poorly absorbed due to their poor lipid solubility. Large molecular weight causes or resulting in poor absorption by passive diffusion. Poor lipid solubility is one of the limiting factor to pass across the lipid-rich biological membrane and ultimately results in poor bioavailability. So many phytomedicines or phytochemicals are limited in their effectiveness because they are poorly absorbed into the systemic circulation when taken through oral route of administration. However, this problem can be solved by incorporating into phospholipids, which can improve the absorption and better utilization of the standardized herbal extracts. So, phytosome process produces a cell-like structure where the herbal extract is protected from destruction by digestive secretion and by the gut bacteria. And these phytozoans have the ability to transform from a hydrophilic environment into a lipid-friendly environment of the enterocyte of the cell. And finally, it reaches into the systemic circulation. So one example is the flavonoid and terpenoid constituents of plant extracts lend themselves quite well for the binding directly to the phosphatidylcholine. So advantages of the phytozoans. So phytozoans, they enhances the absorption of lipid insoluble polar phytoconstituents through oral and as well as the topical route. Thereby it exhibits better bioavailability and they can also result in greater therapeutic benefits. As the absorption of the active constituent is improved, the dose required to produce the therapeutic effect is also getting reduced. So the phosphatidylcholine, which is used in the preparation of phytosomes, beside acting as a carrier, it also has the property of hepatoprotective in nature. So while incorporating the herbal extract with the phosphatidylcholine, it gives the synergistic effect when the hepatoprotective substances are being incorporated into phytosomes. So apart from that, these phytosomes exhibit better stability than the liposomes because the former consists of a chemical bond while it is absent in the later case. And the process of formulation or the manufacturing of phytosomes is relative, a simple process. And whatever the composition of phytosomes, that is the lipid composition, the emulsifier, and other material which are regarded as safe and approved for the pharmaceutical use. And this phytosomes ensures increased duration of action for the phytoconstituents or the phytochemicals. This phytosomes ensures increased duration of action for the herbal drugs, where it also enhances the absorption of hydrophilic polar constituents through the oral route, topical route, and other route, and therefore the viability of the phytochemical is getting increased. The phytosomes produces a little cell where the valuable components are protected from the destruction by the digestive secretion and as well as the gut bacteria. Apart from that, these phytosomes persuades a proper delivery of the phytochemicals or the phytoconstituents to the respective tissues. The nutrient safety of the herbal extracts need not be compromised by assigning the herbal drug by means of phytosomes. And the dose requirement has been considerably reduced because it exhibits maximum absorption of the principal constituents or the phytochemicals. They improve the absorption of biologically active constituents, and also the dose required for the treatment can also be reduced. The chemical bonds which are formed between the phosphatidylcholine molecule and the phytoconstituents shows a good stability profile for the phytosomes. These phytosomes improve the transdermal absorption of phytoconstituents and they are widely used in the cosmetics for better skin penetration and high lipid profile. Apart from this, these phytoconstituents which are present in the phytosomes, they easily permeate in the intestinal walls of the tissues and exhibiting better absorption. The complex which is formed in the phytosomes are biodegradable and the drug entrapment will not be a problem with the phytosomes. They intensify the effect of herbal components by improving or enhancing the absorption process, enhancing the biological activity, and delivering the phytoconstituent to the target tissue. Therefore, phytosomes are considered as a suitable drug delivery system for the herbal preparations. Entrapment efficiency of the phytosomes is high because they have a very good conjugation with the lipids by forming a vesicles. So these are all the advantages we have seen with the phytosomes. Now we look on to the disadvantages of the phytosomes. The main disadvantage which is associated with the phytosomes is the phytoconstituents is rapidly eliminated from the phytosomes if storing is not properly maintained. So these are all the components of the phytosomes. There are three main components which are required for the formulation of phytosomes. The first one is the phospholipids, which could be either phosphatidylcholine or phosphatidylserine. This phosphatidylcholine if you look at the structure, it is a bifunctional compound possessing a lipophilic phosphatidyl moiety and as well as a hydrophilic choline moiety. So one is lipophilic in nature, another one is hydrophilic in nature. And we have to use the organic solvent, that is dioxone or acetate, that is aprotic organic solvent which does not have NH-OH functional groups. And the third one is the phytoconstituents, chiefly flavonoids and as well as the terpenoids. So this is how the phosphatidylcholine orientation happens with the phytosomes. The polar head, if you say, it is having the phosphoester bond. This is the phosphoester bond, and the fatty acid tail is hydrophobic in nature, which would form a bond with the lipid membrane. And this is nothing but the glycerol compounds with the ester bonds. So what is the role of phosphatidylcholine in phytosomes? So phosphatidylcholine, PC, in short we call it as PC, which is an important ingredient in the phytosome formulation to make the phytocomponents compatible with the other ingredients. The phosphatidylcholine is obtained from soya, so which is nothing but it also helps as a molecular building block of the cell membranes and has the property of miscibility with both water soluble and oil environments, and in turn it helps in the absorption of the phytoconstituents by oral route. It enhances the absorption. So the chemical analysis indicated that the phytosomes is usually a phytoconstituents molecule which is linked with at least one phosphatidylcholine molecule. We'll see in the coming slides. So the phosphatidylcholine renders passive carrier for the bioactive phytoconstituents of the phytosomes, but itself it acts as a nutrient, that is bioactive nutrient. It has been proven its clinical efficacy in treatment of liver diseases, alcoholic liver steatosis, drug-induced liver damage, and as well as hepatitis, etc. So the phosphatidylcholine bears two major groups. Those two groups are phosphatidyl group, which is lipophilic in nature, and choline group, which is hydrophilic in nature. Therefore, the dual solubility nature of the phospholipids makes it as an effective emulsifier. So what happens in phosphatidylcholine? The choline portion binds to the herbal extracts while the phosphatidyl group covers the phytoconstituents like a cell form. Further, it protects the active phytochemicals or the phytoconstituents being destructed from the digestive juices. Hence, the phytoconstituents produces a lipid compatible molecular complexes with the phospholipids. Another study has revealed that the choline molecule in humans improves the memory function and also it helps in the muscle control. So coming to the properties of phytosomes. So phytosomes is nothing but a complex between a natural product and a natural phospholipid. So this results from the reaction of hydrometric phospholipids with selected phenol compounds in a nonpolar solvent. And the phytosomes can accommodate the active principles that are anchored to the polar head of the phospholipids, and thereby it forms the integral part of the membrane. These phytosomes are lipophilic substances with definitely metabolite and freely soluble in nonpolar solvents and moderately soluble in fats. So upon exposure to hydrophilic nature or water, they resemble like micellar shape, forming the structure that are similar to the liposomes, exhibiting fundamental differences. So what is the need for the use of phytosome complex? These phytosome complex exhibit a dramatic enhancement of the bioavailability of botanical extract due to their complexation with phospholipid, and thereby it improves the absorption of the phytochemicals in the intestinal tract. These phytosomes have the ability to produce like little cell, and where the herbal constituents or the herbal extracts are being protected from the undesirable environment. Apart from that, this phytosomes shows better stability profile because the chemical bonds are formed between the phosphatidylcholine molecule and as well as the phytoconstituents. Because of that, they exhibit better stability as compared to liposomes. These phytosomes can render a cost-effective delivery of phytoconstituents and also promote synergistic benefits when they have used in the cosmetics to protect the skin against the exogenous and endogenous hazard in normal and as well as in the structural condition, that is stressful structural conditions, environmental conditions. The entrapment efficiency of the phytosomes are found to be high, and they can be used for the enhanced or increased penetration of drug through the skin by transdermal and dermal delivery. Apart from this, these phytosomes are also superior to liposomes in skin care products. products. They exhibit low risk profile, and phytosomes have highly attractive market profile products for the proprietary technology. And this is how the mechanism behind the drug delivery from phytosomes to the cell membrane. So phytosome delivery form, it approaches the cell membrane where it passes through the mechanism of diffusion. So these are some of the differences between the phytosomes and as well as the liposomes. So in case of phytosomes, the active phytoconstituents or the phytochemicals are anchored through the chemical bonds to the polar heads of the phospholipids, whereas in liposomes, the active chemical constituents are dissolved in the medium of cavity or in the different layers of membrane. There is no chemical bonds are formed. Generally, the ratio of phosphatidylcholine to phosphoconstituents is 1 is to 2 or 2 is to 1 in case of phytosomes, but whereas in liposomes, there are thousands of phosphatidylcholine molecules surrounds the water-soluble molecules. And this is the diagram of phytosome and as well as the liposome. So in case of liposome, the water-soluble molecule is entrapped between the phosphatidylcholine drug complex, whereas in liposomes, it is there in the lipid point. Now we will move on to the formulation aspect of phytosomes. This can be done in the laboratory itself. We need to take the phospholipid, and that phospholipid should be dissolved in a suitable organic solvent which contains the drug extract. And this is subjected for the drying process for the formation of thin film and followed by the hydration process. So the solution of phospholipid in organic solvent with the drug extract will be obtained, which can also lead to the formation of phytosomal suspension. Then further, we can isolate the phytosomes by precipitation technique, followed by lyophilization and freeze drying to get the dried product. And this is how the formulation of phytosomes takes place. So polyphenolic compounds with anaerobic solvents by the process of refluxing and concentrating under the reduced pressure, followed by the drying process, causes the phytosomal preparation. So polyphenol with aprotic solvent and as well as polyphospholipid, we have to go for the refluxing process, followed by concentration under reduced pressure, and drying and grinding leads to the formation of the phytosomes. So coming to the characterization of the phytosomes, The first one is the visualization. So visualization can lead to the determination of size and shape of the phytosomes. So this is being done by two techniques, namely the transmission electron microscope and as well as by the scanning electron microscopy method. The next technique is entrapment efficiency, that is to determine how much phytoconstituents or the phytochemicals are being entrapped into the phytosomes. So that can be measured by the ultracentrifugation technique, and we can also measure the transition temperature by using the differential scanning calorimetry technique, that is mainly done for finding out the transition temperature of the vesicular lipid systems. Then we have the surface tension activity measurement. So surface tension activity of the phytosome in aqueous solution can be measured by ring method in Dionoy tensitometer, ring tensiometer. Then stability of the vesicles can be measured by assessing its shape and as well as the structure over a period of time. And the mean size is measured by dynamic light scattering technology. And also, if there is any structural changes, that can be monitored by the transmission electron microscopy. The last one is the drug content. So the amount of drug or the phytochemical can be quantified by modified high performance liquid chromatography method or suitable spectroscopic method, which includes UV method as well. And these are all the spectroscopic evaluation to confirm the formation of a complex or to study the reciprocal interaction between the phytoconstituent and the phospholipids. Various spectroscopic methods have been adopted. So the first one is HNMR technique. So in HNMR technique, we can find out the signals from atoms in the complex, especially with the phospholipids NCH3 groups of choline, which undergoes a uplift shift. Similarly, using CNMR technique, we can identify the broadening and shifting of signals with regard to glycerol and choline upon heating to 60 degrees centigrade. And the other technique is FDR technique. So to compare the spectrum of individual components and the complex formation. Apart from that, it also helps in the stability of phytocomponents microdispersed in water and incorporated into the gel, especially for the cosmetic preparations. Then we have in vitro in vivo evaluation. So suitable models of in vitro and in vivo evaluations were selected based on the therapeutic activity of the phytoconstituents which are available in the phytosomes. For an example, in vitro anti-hepatotoxic activity can be assessed by the antioxidant and free radical scavenging activity. For assessing anti-hepatotoxic activity in the in vivo model, the animals can be treated against thiocitamide or paracetamol or alcohol-induced hepatotoxicity can be examined. For skin sensitization and tolerability studies of glyceric acid phytosome ointment, a commercial product described the in vivo safety evaluation methodology. And pharmacokinetic study. The bioavailability study can be performed using dog as a model to establish the pharmacokinetic data. So one example is silivin phosphatidylcholine complex. And coming to the phytosomes in cancer therapy. So these are all the different phytosomal formulation, that is chlorine paniculatum phytosomes. They exhibited potent activity against cancer cells, breast cancer cells like MCF7. Agial marvelous phytosomes, they exhibited good antioxidant, antiproliferatic, and anti-cancer activity. Luteolin phytosomes, they inhibit NRF2 and sensitize cancer cell to chemotherapeutic agents. Apart from that, luteolin phytosomes in combination with doxorubicin, they exhibited highest percentage of cell death in MDA in V2331 cells. So these are all some of the marketed formulations which are available for the phytozoans. So you can see one by one. Silibin phytozoans, where silibin is obtained from Silimarin marinum, the indications for which it has been used, that is, they exhibited better antioxidant, hepatoprotective activity, and also they serve as a nutraceutical. Likewise, Ginjoba phytosome, Gingoba flavonoids from Gingoba bibiloba, they protect brain and vascular lining. Green tea phytosome, which chiefly contains epigalloctin 3-O-gallot camellia silences, they have very good anti-cancer and antioxidant activity. Olive oil phytosomes mainly contains the polyphenols from Olea idopaceae. They exhibited antioxidant and anti-inflammatory activity. So I have listed most of the phytosomes which are commercially available in the market, which you can have a look. And there are some patents have already also been given for the phytosomal formulations. So the first one is the phospholipid complexes of olive fruits or leaves of olive extract. The phytosomal innovation given, they have claimed that it enhances the oral bioavailability of the olive complex. Then cosmetic and dermatological composition for the treatment of aging. So phospholipid complexes stimulates the collagen synthesis and enhances the interaction between the extracellular matrix and as well as the topical development. And another patent is phospholipid optimization dimension beta 4, which improves the skin treatment, and a composition comprising of ginkgo biloba derivatives for the treatment of asthmatic and allergic conditions. They enhance the anti-asthmatic and anti-allergic effect on the suitable models. Then cosmetic and dermatological composition for the treatment of uh, for the topical treatment. What happened is they found to be good for the wound repairing process. Then phospholipid curcumin complex and berberine complex, and they have used for the treatment of drug resistance, and complex of saponin with phospholipid and pharmaceutical and cosmetic composition containing them. This is the title of the patent. The claim what they have made is improved bioavailability and highly lipophilic in nature. So these are all the some of the patents which have been uh, approved or granted for the phytosome formulation containing the phytoconstituents. Now we will move on to the QBD approach. Quality by design is one of the approach where in a shorter period of time we can produce a stable and robust product. So there are some phytosomal formulation where this QBD approach has been implemented, and I have given some research outcomes. So the first title is development and optimization of epigalloctin 3-gallate nanophytosome using design of experiment and their in vivo anti-inflammatory studies. And this was published in uh, Molecules by Mohammed et al. In 2020. And in this study, they have used two levels, that is low and high, and four factors they have used for the study. One is rate of addition, stirring speed, stirring time, stirring temperature. And desired outcome, they want to get average particle size low and the drug loading expected to be high. So let's move on to this. So these are all the process parameters. So the process parameters, they have used full factorial experimental design. The addition rate is, in case of high, 20 seconds and 2 ml per second. whereas in low it is 60 seconds, 0.67 ml per second. The stirring speed in case of high is 1400 rpm, and whereas in low it is 700 rpm. The stirring time in case of high is 30 minutes, and in case of low it is 15 minutes, and stirring temperature 24 degrees centigrade, 10 degree in case of high, and 12 degrees centigrade in case of low. So the quality based approach for material attributes. So drug concentration in case of high, they have added 100 mg, and low level they have added 50 mg. Phospholipid concentration in case of high it is 100, and in case of low level it is 10 mg. And cholesterol concentration in case of high 10 mg, and in case of low it is 2 mg. Buffer pH is 7 in case of high, and in case of low it is 5.5. So these are all the different experimental parameters. So they have run 16 runs with the different addition rate, stirring speed, stirring time, and stirring temperature, which comprised of high, low, high, 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 high. Likewise, drug concentration also you can see for the material attributes. So in some cases the drug concentration is high, some cases the concentration is low. Similarly, phospholipid concentration, cholesterol concentration, buffer concentration also, it was changing from high to low. That is, in case of buffer it is 7.0 and 5 like that. And these are all the different design of expert for process parameters. So this is what already being explained in the previous slide, and they have observed the different particle size and as well as the drug loading efficiency with different DOE parameters. And DOE for the material attributes also, they found out that using the different combinations of using the fixed concentration, fixed combination, and different combinations, they can find the different particle size and as well as the drug loading efficiency. So the first outcome is the particle size of the phytosomes with green tea extracts were in the range of 100 to 250 nanometers. This can be measured by using the dynamic light scattering technology. The batch 15 showed the highest average, showed the lowest average particle size with an highest entrapment efficiency, and the polydispersity index value of the phytosomes were lower, which is indicated that they are in the monodispersed system with a value of 0.196. So this is the value of the PDI, and they have concluded that the addition rate and stirring temperature found to be the most significant factors that affects the particle size of the phytosomes. So the batches processed with highest addition rate exhibited the smaller particle size of 135 point plus or minus 6 nanometer as compared with the other batches with low addition rate of 1596 nanometers. So other impact on the processing parameters are the addition rate and as well as the stirring temperature played a vital role in the particle size of the phytosomes. So batches which is formulated with highest addition rate exhibited particle size of smaller in nature as compared with the low addition rate. So they have observed that the stirring temperature on the prospective batch exhibited low average particle size. The stirring temperature also has an significant effect on the phytose constituent loading in the phospholipid, where the P value is less than 0.001, and the phytosomal batch processed at low temperature offered high loading efficiency of 87%. So this is the Pareto chart on the main effect and as well as the process parameter on the average particle size. And this is a Pareto chart on the main effect on the process parameters on the drug loading efficiency, which I have already discussed with you. And these are all the interaction of process parameters on the particle size and as well as the drug loading, etc. So this is an interaction of process parameters on particle size and drug loading. There is a two-way interactions they have observed between the process parameters which were not significant on the average particle size of the phytosomes, but they have exhibited significant impact on the effect of drug loading due to slow addition rate and slow stirring temperature. So this was one of the research findings where they have implemented QBD for the phytosomal formulation. So another research outcome is development and characterization of nano drug delivery systems containing versus of phospholipid complex to improve bioavailability using quality by design approach. So this is published in Research in Pharmaceutical Sciences. So they have used Box-Benken design, 3-factorial design using design expert software to optimize the phytosomal formulation. So the independent variables are critical material attributes, which may be low, medium, and high. And they have done the phosphatidyl, they have kept the phosphatidyl concentration denoting X1, stirring temperature X2, stirring time X3. And the responses, that is critical quality attributes, are percentage yield, particle size, and entrapment efficiency. So they have done the box-Benken. Using the box-Benken design, they have prepared the phytosomal formulation. So the independent variables, 3 levels they have used, minus 1, 0, plus 1. So the phospholipid concentration which they have used is 1, 2, and 4 grams respectively. Stirring temperature 40 degree, 50 degree, 60 degree. Stirring time 60, 120, 180 minutes. And the independent variables are the percentage yield should be more than 95%. The particle size should be less than 300 nanometers. The entrapment efficiency should be more than 95%. And they have done 15 runs using the Box-Benken design. And these are all the various responses they have obtained with regard to the critical variables and their outcome. So response value experimental runs in Box-Benken design. They have done 15 batches with three center points, and based on the results, the response surface analysis were carried out using the contour plots, and 3D response surface plots were constructed to establish and understand the relationship of variables with their interaction with other variable. The effect of critical material attributes and critical quality attributes was analyzed, and second order polynomial models were established and adapted into multiple linear regression models. And they've also used ANOVA to corroborate the design. Quality attributes were set at target levels, and desirable formulation design was resolved to apply the checkpoints analysis and desirably approach using design expert softwares. There are some restraints for quality attributes were set at target levels, and desirable formulation design was resolved to apply checkpoint analysis and desirability approach using design expert software. So optimum formulation was designated based on the numerical optimization process using desirability function. So this is the perturbation graph for the CMA and CQA. So once we are increasing the critical material attributes, we can find out its effect on the critical quality attributes. And this is the counter plots for evaluating the critical material attributes on the critical quality attributes with respect to percentage yield, particle size, and as well as the entrapment efficiency. And this is the 3D responses plots for evaluating the critical material attributes or critical quality attributes in terms of percentage yield, particle size, and as well as the entrapment efficiency. And these are all the 3D response plots for evaluating the critical material attributes on the critical quality attributes. So the first one is the yield. The phytosome yield was enhanced with a decrease in phospholipid concentration and at the low temperature. So upon increasing the phospholipid concentration, the yield was decreased at low temperature, and they have evaluated the particle size, which was decreased with low concentration of phospholipid at high temperature. And the next parameter which they have evaluated is entrapment efficiency. There is an increase in entrapment efficiency with low phospholipid concentration at low temperature. However, the stirring time does not influence on any effect on the yield entrapment efficiency, but the longer stirring time exhibited reduction in the particle size. These were the observations made by them. And the numerical optimization of the phytosomes. So the numerical optimization revealed that the phytosome formulation of extract and phosphatidylcholine in the ratio of 1:1 at addition rate of 47.9 with a stirring temperature of 146 degrees centigrade over a period of 120 minutes, which has achieved the overall desirability of 0.94. So value less than 1 is good. The optimized formulation showed the percentage yield of 95.89 percentage with a particle size of 231 nanometers and 95.48 percentage of entrapment efficiency with the predicted values. So the optimized formulation showed the results which I have already discussed in the earlier slides. So these results were also consonance with the predicted values. So these are all the desirable values which is less than 1. And outcomes of this research work based on the implementation of the QPD technique for the optimization of phytosomes, the formulation which contains the phytocomponent with a phospholipid ratio of 1:1 was found to be an optimum formulation. The usage of box Benton design, a response surface methodology or a RSM was employed for the statistical optimization, which involved in the determination of critical material attributes and as well as the critical quality attributes. The interactions of phospholipid concentration, stirring time, and all the stirring temperature were shown in the above figure 3. The optimized phytosome established the maximum percentage yield, minimum particle size, and maximum entrapment efficiency. The particle size, which was found to be 231 nanometer with a zeta potential of 23.2 millivolt, indicated the phospholipids involved in the complex formulation, which may expect to enhance the stability of the optimized phytosomes. So this was the second research outcome. Now we'll move on to the QBD-based approach for phytoconstituent research outcome 3, where they have used the novel umbelliferant phytosomes development and optimization using experimental design approach and to evaluate the phytoprotective and antioxidant activity. So this is published in IJPPS in the year 2016 by Puranik et al. So they have done the optimization using 3 factors, 3 levers employing box-Benton method. The independent variables which have been employed are the umbelliferone phospholipid ratio x1, process temperature x2, and the reaction time x3. For the 2 independent variables, normally the complexation rate y1 and partition coefficient. So they have prepared around 15 batches were prepared and evaluated for the complexation rate and as well as the partition coefficient. So this data was statistically analyzed and validated by the design expert using the ANOVA and as well as the polynomial equations to find out the optimized set conditions. The optimized batch was selected based on the desirability functions. Various 3D responses on surface graphs and contour plots were also estimated. So this is a quadratic equation which has come out of that responses, where y is the measured responses for batch 1 level combination, b0 is the intercept, b1 to b33 is the regression coefficients, and fig d is the weight observed from the experimental values of y, x1, x2, x3 are coded levels of independent variables. So the independent variables, they have done low, medium, and high. Amplifier to phospholipid combination 1:1, 1:2, 1:3. That is low, medium, and high respectively. And temperature 40 degree, 50 degree, 60 degree. And time is 1 hour, 2 hour, 3 hour. The dependent variables are complexation rate, maximum complexation rate, and partition coefficient minimum. So this given the various outcomes, the optimized batch was found by evaluating two responses, that is complexation rate y1 and partition coefficient. The desirability of the optimized batch, that is formulation 4, was maximum 0.992. So these are all the regression equation which was fitted into the quadratic model as given here, and they have done around 15 formulation with the different composition and all to find out the maximum complexation and partition coefficient. So this is the contour plot and 3D responses for complexation rate and as well as the partition coefficient between the materials. So the amplifier phospholipid complex was prepared using the solvent evaporation method, using the Box-Minton method. The quadratic equation which was used for the each response obtained by statistically significant coefficient, that is p-value is equal to 0.0405 for y1 response and p is equal to 0.0483 for y2 were included, and a positive coefficient indicated that the response is favoring the research, while the negative value indicated an inverse relationship between the factor and as well as the response. So it was clear from the above equations that amplifier phospholipid ratio x1, temperature x2 had a positive relationship, and time x3 had inverse relationship on complexation rate, which is nothing but y1. On the other hand, the temperature and time had a positive relationship, and phospholipid amplifier ratio x1 had an inverse relationship on the partition coefficient. So all these things can be observed through the graph which has been shown in the earlier slide, and observed and predicted values for the optimized batch. So the response is complexation rate. So the predicted value is 0.9994. Observed value for the optimized batch F4 is 99.957. Partition coefficient, the predicted value is 7.98, whereas the observed value is 6.52, which are close to each other. Another research-based outcome is optimized acrathine phytosomes which exhibit enhanced cytotoxicity and apoptosis-inducing activities in ovarian cell cancer. So this was also published in Pharmaceutics by Nabil et al. In the year 2020. So phytosomes are identified for their potential in delivering the phytoconstituents for the cytotoxic purpose. The purpose of this study was to determine the potential enhancement of ecatherine cytotoxicity activity for the OVCIR ovarian cancer cells through the formulation of phytosomes. So ICA phytosomal formulation was optimized using box Benton design, where the particle size, shape, and in vitro drug release were used to characterize the optimized formula. The optimized formulation exhibited enhanced in vitro drug release and also uh, enhanced cytotoxicity against ovarian cancer cells. So the design of experiment was as follows. A three-factor box-Benton experimental design was employed to formulate ICA phytosomes. The independent variables are ICA to phospholipid molar ratio X1, process temperature X2, reflex time in hours X3 constituted the independent variables. The responses are the vesicle size Y in nanometer. So these are all the levels which they have used: low, medium, and high. So in low, they have used, that is X1, they have used ecatherine phospholipid 90H molar ratio 1:1, 1:2, 1:3 for medium and high. Temperature 40 degree, 50 degree, 60 degree. And X3 is the reflex time 1, 2, 3 in hours. Respond desirably constant. The vesicle size in nanometer should be minimum. That was their expectation. So these were all the experimental runs they have done. They've got the R square value of 0.9884 with the adjusted R square value, and the p value is lack of it 0.0368, and they have correlated the predicted and actual value, which is having the good correlation among them. And the statistical output with regard to the 15 runs with the different ICA phospholipid molar ratio, different temperature and reflex time, and the vesicle sizes were shown here. They have shown the vesicle, different vesicle size with regard to this quality medical attributes and uh, variables. Then they have done the data analysis. The data analysis using ANOVA showed significant effect on the linear terms, that is ecatherine phospholipid uh, X1 temperature and X3 reflex time on the vesicle size, which is P is less than 0.05. In addition, the quadratic term X12 corresponding to the isophospholipid 90 molar ratio was significant, same significant level. The effects of independent variables on the vesicle size. A negative slope was observed for both variables X2 and X3, indicating an inverse relationship between the variable and the response. However, a positive slope was observed for the variable X1, confirming that higher ICA to phospholipid molar ratio causes increase in the vesicle size. The positive sign of the linear term x1 and the negative sign of the linear term x1 and x3 confirms the observed trend. And these are all the concentration. The main effects of ICA phospho 90H molar ratio x1, temperature, and as well as the reflex time on the vesicle size. So when we increase the concentration of ICA phospholipid, it increases, the vesicle size increases, and whereas when we increase the temperature, the effect of particle size decreases, and reflex time almost no effect on the reflex time in hours. So the 3D response surface plots for the effect of investigated variables on the vesicle size were illustrated that the ICA phospholipid 90H molar ratio exhibited the most significant effect on the vesicle size. So when the concentration were found to be increased, it can also have an impact on the vesicle size. This observation is supported by the highest coefficient of the term x1 in the generated equation. The observed increase in the vesicle size with increasing ICA phospholipid 90H molar ratio could be attributed to the increasing phospholipid content in the phytosomes. So this is a three-dimensional surface plots for the effect of ICA phospholipid 90 molar ratio x1, temperature x2, and reflex time x3 on the vesicle size of the ICA phytosomes. So optimization of the ectherin phytosomes. So the optimum values which is given by the software is 1 is to 1.07 in case of ectherin phospholipid ratio. The temperature is 58.70 degrees centigrade x2 and reflex time x3 is 2.67 error. And these are all the predicted value for the vesicle size 98.25. Observed value is 104.15 with an error of 6.01 percentage. So conclusion of the study. The box beacon is an independent, rotatable, nearly rotatable three-level response surface design that is widely used for the optimization in the pharmaceutical research. So in this study, a three-level box beacon design was applied for the formulation and optimization of ectherin phytosomes with a minimized vesicle size. The independent factors selected in the present study were previously established to influence the phytosomes formulation. These combinations at center midpoints of edges of the design space represents the composition of the experimental runs. The adequate model fitting of the vesicle size data was quadratic model with the highest correlation efficient of R-square value. In addition, the values of the predicted and adjusted R-square value were in good agreement, ensuring the validity of the model employed. The box beacon design was successfully employed to optimize the ICA phytosomes with minimized vesicle size. The optimized phytosomes are almost spherical in shape. So this was the observation observed by the authors. And coming to the last QPD based approach for phytosomes, there's this fifth research outcome. So phospholipid complex loaded self-assembled phytosomal soft nanoparticles, evidence of enhanced solubility, dissolution rate, X5O permeability, oral viability, and antioxidant potential of magniferin. So this is done by Telangi et al. Published in the Journal of Drug Delivery and Translational Research in 2020. So the magniferin phospholipid 90H complex was produced by solvent evaporation method using the central composite design. The optimized MPLC was converted into MPLC SNP using the nano precipitation method. Therefore, a central composite design was used and formed a stable MPLC with the optimal values of 1 is to 1.76, 50.55 degrees centigrade, and the time for the process is 2.02 hours respectively. They have used the central composite design. The MPLC formulation was optimized using CCD to obtain precise information on the formulation process variables with a reasonably lower number of experimental run or trials. So in the present study, three independent variables, namely MGN phospholipids ratio was X1, that is weighted to weight, reaction temperature is X2 degrees centigrade, and reaction time is X3 hours respectively. The extent of complexation rate or yield is percentage was chosen as independent response or variable. The detailed analysis has led to 20 possible runs. So these formulations were prepared and assessed one by one for the dependent variable, which is nothing but percentage yield. So upon fitting the data, a statistical model, a polynomial equation along with a desirability plot was obtained. The model listed the coefficient of the variables and the quadratic and interaction terms having significant influence on the dependent variable. So this is the coefficient of variables on the quadratic model, where they have done 20 experimental trials, and they have found out the interaction between the other variables. So these are all the coded levels and real values for the independent variables. So independent variables are the MGN phospholipid, MGN is to phospholipid 90 ratio, reaction temperature and reaction time, and the dependent variable is nothing but the extent of complexation or the percentage. With these many things it has been performed, and these are all the extent of completion with 20 runs with changing the variables that has been depicted in the table. Encoded levels and real values for each independent variables have been studied. So counter plot for MGN and phospholipid ratio. So at a fixed MGN phospholipid 90, there is an increase in the yield. Later, X2 and X3 increases to a middle level. The yield is also getting decreases. So yield comes down. Then effect of the reaction time. At a constant reaction time, the yield was found to increase. So this is nothing but the yield was found to increase due to increase in the factor x1 and x2, and middle level then decrease the yield. So this is nothing but decreasing. Then effect of reaction temperature. At a fixed reaction temperature, the yield was found to be increased due to increase in x1 and x3 to middle level. Then there was a decrease in the yield was observed. Then we have a 3D response surface plot where we can find the effect of MGN phospholipid ratio. Also at fixed MGN phospholipid ratio, the yield also increased as X2 and X3 factor increased to a middle level, then followed by decrease in the yield. Then effect of reaction temperature. At fixed reaction temperature, the yield was increased by increasing x1 as well as x3 to the middle level, then the yield found to be decreased. Then effect of reaction time, as seen in this figure, at constant reaction time, the yield was found to increase as the x1 and x2 increases to middle level, then followed by the decrease in the yield. So this is with regard to effect on reaction time. Then desirability, the analysis of the design provided one possible solution for the experimental trial that showed the recommended values of the independent variables. So the desirability value is near to 1, which is good enough. Then contour overlay plot based on the overall combination of the desirability and design space provided the following optimal values for the independent variables such as x1, x2, and x3. So x1, it is 1 is to 1.76 with regard to MGN phospholipid ratio, weight by weight. Temperature is 50.55 degrees centigrade, and time is 2.02 hours. So the validation of the model. So central composite design generated a model. Additional trial formulation using optimal values have been carried out. Actual yield is 90.35, which is close to the predicted value of 90.14. So this led the experimental confirmation with the validated robustness of the design generated by the quadratic model. So the predicted error was found to be less than 3%. So another QPD-based approach for phytosomes research outcome 6. So this is the formulation and characterization of abiganin phospholipid phytosome for improved solubility, in vivo bioavailability, and antioxidant potential. So this is published in the European Journal of Pharmaceutical Sciences in 2020 by Telange et al. So abiganin is a hydrophobic polyphenolic flammide. Generally, it is indicated for its antioxidant, antiviral, antidiabetic, tumor inhibiting activity. So the clinical potential of abiganin exhibited its poor aqueous solubility that is less than 2.16 microgram per ml, and also it also undergoes rapid metabolism and as well as low oral bioavailability. So the design of experiment was full factorial design was implemented to understand the overall influence of independent variables. So the first variable is X1, drug to phospholipid ratio, weight by weight, temperature X2 degrees centigrade, extent of epigenetic incorporation, which is nothing but percentage yield. The two independent variables X1 and X2 were selected at three levels, namely -1, 0, and +1, that is low, middle, and higher, resulting in 3-degree factorial design of 9 possible combinations. Yield was defined as the dependent variable in the following polynomial equations. So where y is the dependent variable, b is the coefficient of independent variable x, the main effects x1, x2 represent the possible aggregate effect on both factors as they change independently from their lower to higher level. The interaction term x1, x2 showed how response changes when two factors are simultaneously changed. The polynomial term x12 and x22 describes the non-linearity. And this is different coded levels and real values for each factor that has been studied. So the level values were 1:1 for epigenin phospholipid ratio, 1:1 to 1:3, and reaction temperature from 40 to 60 degrees centigrade, and the dependent variable is extent of epigenin incorporation or the yield, percentage yield. So totally 9 runs have been done with different variables, and these are all the extent of epigenin incorporation or percentage yield in terms of weight by weight. So this picture shows that changing different factors influence on the extent of yield and on the extent of epigenin incorporation or the percentage yield. So this is the response surface plot on the entrapment efficiency. So 9 experiments were performed using 3-degree full factorial designs. The results indicated that the extent of epigenin was significantly influenced by two variables, namely x1 and x2. The extent of epigenin incorporation was ranged from 67 to 94 percentage. So these data were utilized to construct a quadratic model, and this is the polynomial equation which is used for the deriving the conclusion. The coefficient values such as b0, b1, b2, b3 were statistically significant where p-value is less than 0.05, whereas b4 and b5 values were found to be non-significant in nature. The counterplot on entrapment efficiency. A quadratic model was employed for the experiment. The optimum values for the value for the variables are x1, weight by weight, 1:2, reaction temperature x2, 60 degree centigrade. Coming to the conclusion part, the phytosomes connects between the conventional delivery system and novel delivery systems. Phytosomes are the technically advanced form of herbal extract that are better absorbed, which results better than conventional herbal extract, thereby improving the pharmacokinetic and pharmacological parameters, which can be an advantageous for treating various diseases. The nutraceutical products based on the phytosomal technology become available at the site of action, especially in case of liver, kidney, brain, heart, etc., with less dose as compared to the conventional plant extract. And the phytosomes have wide scope in cosmetic technology, cosmetics, and many areas of them are revealed to be future in the prospect of pharmaceutical applications. Phytosome products offer extemporaneous applications in cardiovascular, anti-inflammatory, anti-cancer activities. They play a vital role in efficient herbal drug delivery of hepatoprotective phytoconstituents, namely flavones, xanthones, and terpenes. Phytosome products show their potential in cosmetics, especially in the areas like anti-skin aging and non-pathogenic skin conditions. And these are all some of the references which are related to this presentation. Thank you. Any queries? I'm happy to answer. Thanks for the patient hearing. Thank you, one and all. Hello?
Sir, sir.
Over, sir.
Okay, sir. I am waiting for any queries you are asked.
Yes,
If
sir.
anyone is asking, I am looking for that one.
Yes, sir.
Okay, sir. There is no questions from the chat box.
Yes,
Sir,
sir.
thanks for your presentation, and that presentation was very nice presentation. And you also given the most important topic, that is the phytosomes and their importance in the present scenario and the drug discovery, that is from the herbal side.
Yes, sir.
Thank you, sir. And their importance, phytosomes and quality-based development about the phytosomes and the importance and role of phytosomes in the formulation development and in the various formulations.
Yes, sir.
Okay, thank you, sir. And one question is raised here: stability issue of phytosomes in lab.
Yes, sir. Actually, if it is not stored in a proper condition, there may be a leaching of phytoconstituent from the phytosomes. So care must be taken in such a way that it should be stored in an appropriate conditions. Refrigerated conditions is ideal, sir.
Okay, thank you, sir. Thank
Thank
you
you,
very
sir.
much for all the participants. And small announcement: tomorrow session is starts at according to the Indian time 3 p.m. That is afternoon time. 3 p.m. at the session is start tomorrow. Thank you all. Thank you, sir. I will in touch with you.
Thank you so much, sir.
Thank you, sir. Thank you very
At
much.
this juncture, I thank the management of CHIPS for giving me an opportunity to discuss these phytosomes with the fraternity faculties and other
Thank you, sir.
experts.
We always welcome to you from the JSS UT or JSS Mysore. We are
Thank you so
always
much.
welcome. Thank you, sir. Thank you very much for your
Thank you,
nice
sir.
program.
Thank you, one and all. Have a nice day. Have a nice weekend.
Thank you, sir.
Thank you so much.